Good morning. Thank you everyone for attending this KOL call, which serves as a follow-up to our 6th annual Genetic Medicines and Cell Therapy Manufacturing Summit that took place last week. My name is Keay Nakae. I'm one of the senior healthcare research analysts at Chardan, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. Before we get started, our compliance department has asked us to read this statement. Our participant has attested and agreed that they will not share or discuss any material, non-public, or confidential information that they are aware of. The recording of this call will be made available to clients after the event. Joining me on the call this morning as co-host is my colleague, Senior Healthcare Research Analyst, Dr. Geulah Livshits, and our special guest, Dr. Donald Fink, Master Practice Expert Regulatory, Dark Horse Consulting. During the call, for anyone in our audience who'd like to submit a question to ask of Dr. Fink, please type it into the box and we'll try to ask it on your behalf. At this year's summit, we heard from 25 public and private companies regarding the current challenges and advances in genetic medicines and cell therapy manufacturing. Topics included the logistics of incorporating process improvements, clinical holds, comparability, scale-up, supply chain logistics, qualification assays, analytics, new technologies, make versus buy considerations, and to what extent regulatory standards are changing. In this KOL follow-up call, we will discuss a number of these key issues from a regulatory perspective, leveraging Dr. Fink's 27 years of experience at FDA and CBER. So to start, thank you so much for joining us today, Dr. Fink. And before we get into the questions, could you share with the audience your background as it relates to cell and gene therapy CMC?
Yes, I'd be happy to, and thank you very much for this invitation to participate in really what's, I think, a leading edge and front, cutting edge place to be right now in this space of cell and gene therapy. My experience goes back to the Center for Biologics, FDA, where I began in 1993 in recombinant monoclonal antibodies and therapeutic proteins, and then in 1999 transitioned to cell and gene therapies, where I then spent the remainder of my career all the way until 2020, working in the space of cell and gene therapies, primarily in the cell therapy branch within that division, which then grew into an office called now, as you will familiarly recognize, the Office of Tissue and Advanced Therapies. And throughout that entire time, I was in the CMC subject matter expertise area. And if you think about that timeline, that really coincides with a nascent field growing up. And so what my experience is that whole process leading from sort of genesis or first steps into the space for new products all the way through some of the first approvals, which have been primarily successful in the gene therapy side with the ex vivo transduced cells and CAR T cells, but certainly have seen a lot of effort and a lot of experimentation, a lot of clinical studies going on in the cell therapy space for a variety of products from so somatic cells differentiated all the way to pluripotent stem cells as being starting material for the manufacturer. So I have a breadth of experience in that place, and I've covered the regulatory waterfront from the first engagements, which would be interact meetings, all the way through pre-IND, IND, amendments, up to a BLA submission and post-approval activities. So that kind of paints that picture for my experience.
Well, very good. Well, Geulah, let me hand the mic over to you to ask the first few questions.
Great, thanks, Kay. So the genetic medicines field has seen a number of programs being placed on clinical hold by the FDA over the recent years. Some of these are related to safety and some relating to CMC issues. So can you provide us with your perspective on why we're seeing some of these holds?
Yes, I'm happy to do that. I think there's a variety of factors that can be teased out, but primarily the key, I think, is just activity. There's activity in the space, there's more innovation, there are more trials, there's more exposure, more opportunity, particularly on the safety side, for events to be noticed and seen, detected, and on a first-time basis. So a lot of that is due to success. It's due to success of getting a clinical trial started. It's due to success of exposing patients. And it's due to the fact that there is at least approvals of substance, Luxturna being one, which is just a gene therapy native, but the other being the CAR T manipulated cells, which are considered a gene therapy. And as you get success, you get more players, and you get more players, you get more exposures, and you get more exposures, and you start to see events. And the other thing I would say is, when you're dealing with, and I'm staying on the safety side for the moment, when you're dealing with these safety factors, we know regulatorily that your efforts to weed out safety signals in preclinical animal models is very limited. The animal modeling is not terribly, doesn't with fidelity reproduce human situation, the physiology, the biology, the morphology, all of those things are different, and you're putting by-ask human products, human materials into an animal. So you're not seeing perhaps fully what the likely events are to occur. I think the other thing on the safety side that we're seeing relates to the types of exposure and dosing. And so some of these diseases that are being targeted, even though they're perhaps monogenetic corrections, have global distribution issues. You need to get genetic modified material throughout the body in a systemic way, which means high doses of exposure, which I think in the case of a viral vector, for example, being your transitory delivery system has immunogenic consequences, which are now being unfolded. And so that's part of that safety piece. It's experience, it's exposure, it's a lack of earlier detection in a signaling system, and finally dosing. And all of those come together to form more safety signals being generated. I don't think the safety signals generated are terribly unexpected in the sense that they match what you could have perhaps surmised based on the material and its interaction with physiology, but still you hadn't seen it, and now you are. And so the next step becomes to mitigate those risks as best as possible. On the other side, where you talked a little bit about say CMC. So, well, CMC is obviously linked to that. It's linked to how is the material made, what's brought in to make that material, what contribution can materials in the manufacturing process contribute to, if they're not completely removed or eliminated or integrated in some way, contribute to behaviors of the product. So there's that aspect. But I think on the CM side, primarily, there's a little bit of a disconnect between expectation on the agency side for what's called potency measurements or activity measurements. And the disconnect is in interpretation and in timing. And the timing becomes on the gene therapy space, where there's work in the rare disease area, for which is, it makes sense because you have a single gene defect. Hey, we'll go fix that. It's a target. That's useful. But there are not a lot of subjects or a lot of patients in that area, which means that every patient enrolled in a trial counts, and it counts towards both safety and effectiveness. And in order for that to happen, CMC development needs to be commensurate with almost a late stage clinical trial, because if you want data across the entire collection period to be useful in terms of efficacy, then the CMC has to match that. And here we're having an issue with potency, the status of that assay, what that assay is able to detect. Does the agency believe that it detects something meaningful to clinical effectiveness? And is it established well enough at the point of utility that when your first patient gets treated, you can say that clinical effectiveness data collected can be used throughout the entire process of development all the way up to licensure. So I think that probably in the space is the key stumbling area for CMC. We could talk about comparability as well, and there may be other questions where we get to that, but comparability fits into this scheme because it relates to, right, CMC. If you start at square one, but you start to make some modifications based on your experience, and then you want to introduce those changes, you have to demonstrate similarity between product that was before the change and after the change. Now, we use the term comparability. Comparability really is an exercise originally intended to be performed after a license. And the reason that is you can have a manufacturing process and a clinical indication that have been through the approval system and now are recognized to be the baseline for which you can make other new judgments. Before that, you don't have that luxury of having a vetted baseline. So comparability becomes a difficult issue, and it stymies in some ways innovation, because if once you execute a change, you're obligated to show similarity. And if that bar for that comparability is going from what's expected post-marketing and now brought into the space of pre-market and development, it becomes a really, really difficult exercise in order to get enough data to support a statistical evaluation that meets what the agency is currently asking for, at least in my experience, which is equivalence. And equivalence is a little bit higher in my mind than something that's highly similar. So, but we can have a debate about that. But I think that's where the big stumbling blocks are. So if there's any, I think that kind of wraps that up. We can probably, I could, you know, I could talk about this for all day, but let's move to the next one.
Yeah, so maybe we can come back to comparability in a little bit more detail in a second. But before we go to that, you know, let's back up. Can you discuss the key things that regulators are actually looking for with respect to CMC for both the, you know, the product itself as well as critical components such as vector or editing enzyme, and you know, at different stages of development? Yeah,
we certainly can do that. Let me just do a general description, and I'll maybe talk about expectation related to process control. So process control and demonstration of reproducibility in terms of capability of making your investigational product that's really the primary CMC objective during the investigational stage. Can you manufacture something reliably and consistently? Now, on a product by product, case by case basis, there can be classes certainly of materials, but typically at this stage where virtually everybody coming in has something new, by that I mean it's not been ever marketed before, it's not been ever evaluated before, it's not been ever through approval process before. You're really essentially having to write the story every time you bring a product forward. So to demonstrate that consistency and reproducibility, there's a couple terms. Again, these are probably terms I would say misborrowed, but used from small molecule drug manufacturing or even monoclonal antibody, large capacity, large scale manufacturing, and the concept of quality by design and intentionally creating a manufacturing environment and space where you can have products that can be demonstrated to be under controls because you can evaluate them in a number of reliable and specific ways. And the terms borrowed from that are critical quality attribute of your product, whether it's a gene therapy or a cell therapy. And then based on these sort of features of your cellular or your gene therapy material, you look at things or stages or unit activities within the manufacturing process, which become identified as process control parameters. And so what you're trying to do is identify those things that you, the innovator, view as being necessary for that material to demonstrate in order to meet what it is you intend to give to the subject. So you identify those through your knowledge, through your experimentation. They can be anything from, you know, morphology to enzymatic activity to cell surface markers, attributes of the material that are significant in order to convey a clinical effect. And then you develop a process about which you impose controls that constrain the manufacturing process to meet a product that falls within certain acceptance ranges of acceptability for those parameters that you're measuring or those attributes, and you build that as part of the CMC process overall. So that's sort of the integrity and the space that you're trying to operate in, whether it's cell or gene therapy. That's the overall guise. Now, you asked a specific question related to sort of terminology. And this is falling out a little bit more on the gene therapy side and certainly gene editing space. And the term is a critical component, and I'll come back to that in a second. Want to remember, I think folks maybe on the call may know this, GMP expectation moves a little bit throughout process of developing a product. So overall, there's and if, as a comparator, let's use what goes on in the EMA and in Europe. So to go get a product into a clinical trial in a European center, whether it's an academic sponsor or a industry sponsor, you have to demonstrate sort of manufacturing capability that meets their definition of current good manufacturing practice. And you're inspected by a quality person who goes through and vets that and assures that, so that you enter into the very first manufacturing stage with a quote unquote GMP compliant process. The agency in cell and gene therapy has taken a different route to that. So GMP is always expected in its conceptual design, but verification of that compliance evolves over the development. And in fact, if you read statutory language in a phase 1 trial for a new material or new entity, GMP compliance with GMP is not expected. They're exempt from that. They're exempt from the rigor of what's identified in regulatory statutes for GMP. Having said that, though, they do have what they call phase 1 GMP compliance. So I think this has been sort of the moving target in terms of what expectation is being on agency side for rigor of manufacturing. And now we're going to take that a step back into what we're calling critical components. So in a GMP or in a well-controlled manufacturing process, you bring material into that process, and you do that in an organized way under specific procedures with certain criteria to say, yeah, that material is suitable for purpose. I am now going to enter it into the manufacturing stream. What has sort of happened with that is that there's been this expansion that in certain materials, i.e. critical components, and here gene editing is pretty clear on what are those, which would be like a guide RNA material or the endonuclease material. So these are the things that direct the editing and perform the editing in a well-defined way. They've been identified as critical components, and the expectation now is that those materials, which are not the product per se, but are an integral piece to the product to assure its activity, are manufactured to the highest degree, or the highest quality available. So if an entity goes out and says, hey, I have guide RNA and I can manufacture endonuclease in accordance with the principles of GMP, you now have what the agency views as the highest quality material, i.e. GMP manufactured. Typically, GMP manufacturing is a requirement for an active pharmaceutical ingredient, for a drug substance, and a drug product. None of those mention this word critical component. Critical component is now being layered into, particularly on the gene therapy side, into that place where the expectation is if you can get that material from the earliest days to do the first edits, whether it's the product itself or material that's going to be derivative for that product. For example, you're going to gene edit a cell, which then becomes a master cell bank from which you then produce product. They are still asking, use the highest quality material available, and in their view, that's GMP made material. Now, does that mean you have to only use GMP material? No, you don't. And it's a recommendation, but I can, my experience, at least since I've left the agency just over a year ago, is that to meet the qualification of a non-GMP material is difficult. It's not impossible, but it's difficult. It will take time and resource and effort in order to get that material. So if you've come in with something using a gene edited cell and you haven't had access to GMP I don't want to say a grade. It's not a grade. It's a type of, it's a way the material is made. If you haven't had GMP manufactured materials used there could be some significant challenges to allowing those materials to proceed into the manufacturing process for clinical development. So that, I think, it kind of gives you a broad thing of what CMC is meant to be, and a specific sort of area where we're seeing increased expectation regarding what the agency terms quality for materials used in the manufacture.
Great, that makes a lot of sense. And so, as you mentioned, in terms of comparability, sometimes companies do need to modify their process during the clinical development trajectory, and whether that's for commercial scale-up or to increase the process robustness because they've started with an academic process. So can you expand a little bit on the ways that regulators prefer to see comparability demonstrated?
So they would love to see it in the paper diagram way, which would be perfect, and doesn't happen. But if we were just to there's several. Well, of course, there's several features to this. I think the key feature is where in your development you're trying to introduce a manufacturing change, and that becomes sort of a driver for how difficult a comparability exercise becomes. The other point to it is that I think we touched a little bit on it. It's sort of a unique category to all of this is the pursuit of rare diseases, because rare diseases do not allow for canonical development. So ideas about comparability, regulatory expectations related to comparability, the great recommendations you get through interactions with the agency about comparability are all really premised on the idea that you will be manufacturing multiple products. You'll have multiple manufacturing runs. You'll have a history of information to draw upon in order to reach conclusions about pre and post manufacturing change, similarity of the material, or if you want to say comparable materials. And that's in the rare disease space, this is not a reality. The other place where introducing, so yes, you want to innovate, and clearly if you're not needing to have a large capacity manufacturing process to support clinical trials, you don't do that. You manufacture to the degree that's necessary to support the ongoing activity. Let's assuming that you're now in a space, CAR-Ts are a little bit in this space, right? Even though they may be covered by, in some places, some diseases are still orphan indication, but orphan doesn't mean rare. It simply means below a certain number of patients per year, but that can still be in the thousands of patients, which is a bigger number than the tens of patients. You may need to find, to get out of the clinical trial phase where you're only doing studies with 25, 50 patients, maybe 100, maybe, to now you want to jump to being able to manufacture for maybe 500, 1,000 a year. That's a significant change. And so you need to introduce that. Where would be the best place to do that? Well, you can innovate along the way, but each, as I pointed out earlier, anytime you innovate, if you can't demonstrate comparability in a rigorous way, it jeopardizes any prior clinical information that you may have regarding efficacy in order to be able to pool that data in the end for statistical evaluation. So it depends on how much manufacturing has gone on, how extensive is that experience, and then the rigor with which you can do a post-manufacturing change comparison. And this often is involving statistical plans that have to be considered in advance in order to demonstrate to the level of satisfaction on a purely analytical basis comparability. And if that's not going to be achievable, then you're going to have to bridge between materials. You're going to have to perhaps have a clinical arm in a trial where you use what we call the legacy manufacturing process. First 3 subjects, legacy product. Next 3 subjects, new product. In an arm in a study. So what you're actually doing is you're doing head-to-head collection of clinical data with pre and post materials, showing clinical output to be and safety profiling to be adequately or sufficiently similar, and then proceeding from that point onward with your new material. Again, in a rare disease space, that's not an option, right? It's not like you can transition. If I'm only seeing 1 subject, 2 subjects a year, that's going to take years to do that exercise if I need to do clinical data. It's also going to be difficult to have pooled enough manufacturing data for there to be enough substance for doing a statistical analysis. And one place I can see, and I would discourage folks to do this if possible. So let's say you're getting into your collection. We'll call it. Some people might call it phase 3, but as I said, it's becoming less and less common to have really what's a canonical phase 3 trial now. But let's say a pivotal study intended to support the license, and you're using your updated, we'll call it the updated, that could have undergone some changes to this point, but now you've entered phase 3 and you're using the current updated manufacturing process, and you treat all your subjects, collect all your clinical data, and then before you get your license, you come to the agency and said, okay, here's what we really want you to do now. Here's the manufacturing process that we want to use once we get the license for all the clinical data, and we're going to show it to you now, and we want you to agree to it now without actually treating any patients with this material. Unlikely to happen. That's my experience. Unlikely to be able to get an analytical assessment that's going to pass muster, so that you could simply transition with your clinical data, old manufacturing process, to the new commercial manufacturing process as a post-market exercise, as a post-market treatment immediately. In other words, so you would get the license, you get approved on the old process, but then you would immediately go to manufacturing with the new process. That's unlikely to occur without some sort of clinical data to support doing that. And people are trying to do that, and are finding that to be a difficult place to get to. My advice is more along the line, you still may be obligated to have to do some clinical investigation, but is to get the license and do this all as a post-marketing approval exercise, because it goes all the way back to what I said earlier. You have something that's been approved already by the agency, and now you have a solid baseline to work with. So comparability is difficult. Analytical comparability has become very challenging, and I think the key piece of the analytical, for setting aside all of the various attributes, you can have a number of what you call critical quality attributes, which you may believe to be critical. Agency may not necessarily agree with you. So you want to have agreement with the agency about what actually are the critical quality attributes of relevance. But the most important piece to be able to measure is the potency or activity assay, because really all those other things aren't necessarily meaningful if they don't really convey activity or deflect its activity or modify activity. The thing that you really want to know is that activity assay, the establishment of it, the rigor of that. And if that's a solid, really well recognized, and maybe a matrix of assays, the agency is wholeheartely on board with, bingo, that's a great potency assay, you might be able to get through with an analytical exercise using materials and comparing them directly. And the other thing that the agency is often asking is, when you make a manufacturing change, is you do it in what's called like a split product manufacturing exercise. So you would take the same starting material, and half of it would go to the legacy process and half of it would go to the new process, and then you would manufacture in parallel using two different processes, same starting material, and do your testing and evaluation comparisons on that same starting material. That's not often doable, because often that material, I want to say in certain contexts it's not doable, because that material is coming from, let's say, a patient, and it's in a limited amount, and you have developed a manufacturing process that says, I need all of that. I need all of that material to start the manufacturing process. And now you're saying, okay, we're going to split it into two. Sometimes it's not practical unless the agency agrees that you can use a scaled down version of the manufacturing process, but that has to be agreed to and negotiated. So those are some of the things that impact that comparability exercise. Where is it? What is the magnitude of it? And what does the agency believe needs to be done in terms of bringing in clinical evidence? So.
Great. And so you touched on this a little bit, but do you have any further views on the differences in CMC standards or CMC focus areas for, let's say, IND versus late stage studies and towards the regulatory filings that we haven't already covered?
Fair question. I think the agency's always sort of given you the sense that there's this. First of all, I want to say the end game, the end standards, if you will, the end regulatory expectations aren't different regardless of where you are in the process. What is different is the extent of the information needed to fulfill that obligation. The information burden or evidence burden early development is less than it would be in terms of compliance at the end of the process, but still what you're trying to achieve in terms of demonstrating manufacturing capability, consistency, reproducibility, and in the end, the difference at the end stage, certainly prior to the filing the BLA application, is that you're moving towards higher degrees of what folks refer to as qualification of the process, and B, the assays and the testing. So there's a higher expectation on the performance in ways that are measurable objectively for the manufacturing process, as well as those tests that are used to assure the process is functioning well. So again, you may learn along the way and have to make some adjustments based on experience. Okay, so we found that when we held the material in storage for this long, that was actually a bad idea. Or we found that when we used this process intermediate maintained for under certain conditions, we really needed to fix that formulation of holding, those kinds of things. So there's adjustments that get made during the process. But again, the level of that evidence is lower. So that's the only thing that's lower is the burden in terms of evidentiary. But as you move forward, that becomes a fuller story, needs to be pictured. And by certainly when you get into the BLA space and the license, then everything has to have undergone quote unquote a validation. So the manufacturing process needs to have gone through a validating exercise. As demonstrated by process performance qualification runs using all kinds of controls. And the same would be true for your assays. You would have to submit in that application validation evidence for all of that. So that's really, again, it's just the extent of information and the level of, we'll call it qualification or validation expectation, and that grows over the lifetime. But what you're being asked to do in terms of you need to demonstrate safety and effectiveness, those aren't different standards. Just evidence to support them is different in terms of quantity.
Great. I think I'll pass it back to Keay for some additional questions now.
Thanks, Geulah. Dr. Fink, we've heard sponsors express views that regulators are, to some extent, learning on the fly with respect to different CMC aspects that they think are now more important to focus on as their experience with these emerging cell and gene therapies increases. And this can lead to some complaints that the agency is moving the goalpost on them. So how would you respond to these types of comments?
Yeah. I get that's the way it feels. I think there's no question about that. So let's think about this. What does the agency always say? They always say that they're a science-driven agency that makes its tries to make its decisions based on the best available science. Let's talk about timeline. And I sort of had something. If I don't get back to it, I'll introduce it here. So I think we've all seen when you go into the private side and you look at ganted out timelines for various activities to occur. So if somebody asked the average citizen, how much money need you make to be rich, they'd say about twice as much as they're making right now. I would take every Gantt chart timeline I've looked at for what they expect for development to do and say that, well, you need to multiply that by at least twice and probably more because the regulatory agency doesn't move at your speed and never will. And so I think the expectations about what the agency's timelines are probably misguided. The agency is working deliberatively. It's working in a place of, as you suggested, novel product areas, new materials. And so if that timeline is years to go from start to finish, sometimes, and I'll give an example of one of those. there aren't very many cell therapies approved, and those that are approved, aside from maybe Dendrion's Provenge, really aren't a canonical cell therapy. They're sort of, kind of these, like Carticel was actually already on the market, and then they brought it in to make it licensable. Cord blood is just something that's always been there. A couple of the products that have been licensed are no longer even available commercially. So cell therapy has not been a robust space for product approvals. And an example of timeline, in 1997, the agency was first approached about using pancreatic islets from cadaveric donors to treat diabetes. And these have been shown throughout a long history of time to be actually effective. So let's call that islets. They're like a cell therapy. They've been there, first approached the agency in 1997. The first time an application was submitted to the agency for islets for approval for something that they're known to be able to do was 2017. And that's 20 years. And when that application was turned in, the agency refused to file, adding still more time. So it's taken over 20 plus years for something that even looked established to get to a marketing place. Now, let's use this as an extreme example. So go somewhere in between that. Science is changing in this area very rapidly. New information becomes available. New technologies for querying materials becomes available. Once the agency becomes aware of these types of innovations, and whether it's through informal channels or the formality of another sponsor other than you introducing something, they have an awareness, and they'll look at that awareness and they'll try to understand the significance of having that new knowledge. Is it really valuable? And if it's valuable either for understanding a material aspect of the product or assuring the safety of material, they're going to bring that on board, and that can happen anytime. And I can see how that can be a little bit frustrating if you've been engaged in a manufacturing development process for a number of years, and now suddenly you're being asked to introduce something new that's going to take time and effort to bring on board. So in the sense of a goalpost moving or changing, if that is your definition of a goalpost change, it could be think of the goalpost as being like really wide. The goalpost hasn't moved, but now you've narrowed down into where that target is as to what you need to bring into to meet that requirement. That's another way of looking at it. I do think that there can be a field sense of different expectation based on the rapid pace of science and scientific discovery, and we're all victim to that. And it becomes a case of what can you rely on as precedents, what can you rely on as being foundational for moving forward. And I think that when these new things come on board, you need to have careful conversations with the agency regarding their expectations. And is it really essential? Are all of these things, is all this new knowledge, if it's related in some way to your cellular material in the manufacturing process, essential, or can you continue to proceed along the lines that you have been, and perhaps make that change at some other point in development, and maybe after licensing? So that, yes, I think there is a sense that happens. Yes, I think it is perceived, it's triggered by new information, and yes, I think the agency has a innate curiosity to ask. And so sometimes you have to work through that and negotiate with them. Is this really essential, or can we continue to move forward as we have been? So I would say that way, yeah.
Yeah, well, let's kind of drill down a little bit on conversations between the agency and sponsors, especially the type of communication that occurs, at type A and type B meetings. We know that the FDA provides minutes of its formal meetings to sponsors after the fact, but can you tell us how much detailed feedback the agency actually provides in a meeting? And is there really any reason for the sponsor and the FDA post such a meeting and the minutes not be on the same page regarding expectations with respect to approvable primary endpoints and trial designs? Give us your view on that.
Yeah, a couple places. First of all, I'm going to walk through probably the most common meetings. Let me just start by saying a type A meeting is the least likely meeting you'll ever have. And the type A meeting is largely for stall development and largely for adjudicating dispute resolution. And it's sort of a last gasp effort. And so if I'm on the outside and I'm reading so-and-so went for a type A meeting, I'd be concerned. And my concern would be based on they haven't been able to meet expectation and now they're looking for a way out. So that's an extreme case. The more common meetings are type B, and you have type B as pre-IND meeting, and you have type B as end of phase 2 meeting, maybe end of phase, but certain end of phase 2 meeting, pre-phase 3 meeting. As examples of type B meetings, you have pre-BLA meetings. Those are all type B, and then type C are the more common ones. And type C meetings typically are to query about development in a discipline way. It can be all 3 disciplines of CMC, preclinical or clinical, or it can focus on any one of them. And if you get into the designation categories such as fast track, breakthrough, and on RMAT, regenerative medicine advanced therapies, you're entitled to more of these meetings because the purpose of these expedited pathways is to engage. So the type C meeting is going to be your primary tool. What goes on in that meeting to prepare for that is you prepare a briefing document to submit to the agency with your questions that you wish to address. The agency doesn't want 100 questions. They'll ask you restricted to around no more than 10, 10 to maybe 12, but no more than 10. And folks try to lever that. They'll here's question 1. Subpart A, subpart B, subpart C, subpart D. Agency says, no, those are all questions. So you're entitled to 10 asks. You're going to present a briefing document with information. You're going to state your position, and you can provide substantive information to support your question. So that's what goes into preparing the agency, whether it's a type B or type C meeting, for this interaction. And that's what they have to go on. Then once the meeting is conducted, usually nowadays, most of these are written response only. And so there's the clarity of your question and the understanding of the agency are primary factors that drive the success of what information is provided. What I would like to point out is a great example that I heard about recently. Interact meetings, and remember, these are way early, and they're intended to provide a first look into your product development and try to provide you with guidance about how to do preclinical testing. These are still being granted by teleconference. Now, mind you, only 1 in 3 Interact meetings request is granted, so they're controlling the number of them. But you can still have person-to-person communication with this, and it turns out in the experience that was shared with me, this is extremely valuable. Why was that valuable? Well, so the agency, right, they had the briefing book, they read the question, they read the position, they had the supporting information. When they went to provide their response, they said, no, we don't recommend that, or we don't agree with your position on this. And the sponsor was able to say, well, in your response, it doesn't seem that maybe you, A, we might not have given you something that you needed, or B, you didn't quite understand what we asked. So they had a dialogue. And once they had that dialogue, the agency said, oh, now we get it. Okay, yeah, no, that makes sense. Yeah, now what you said makes more sense. That's not happening in written responses only to the same degree. That part of back and forth is missing, and that can be problematic. Now, you do get your written responses, and they spend a lot of, they spend a fair amount of time doing that in order to provide as much clarity. But again, it's based on their understanding of what you provided, and they may have missed something. Now, once you get the minutes, you are entitled, well, I don't say entitled, but you are allowed to seek clarification based on the written responses. So you can go back, but again, this is in a written sort of exchange. We're not on the phone. We're not having face-to-face. We're going back. With regard to your response to our question number 2, we want to point out this following feature. Now, you can only clarify. You can't add new. You can't bring in new information. If you bring in new information, they won't address. They'll return it back, and they won't even address that. So I think the aspects of number of meetings available if you're not under a designation, the fact that they're written response only, and the fact that you don't have, that there can be some disconnect between what the sponsor tried to convey, what the agency understood being conveyed, and then the response to that. You can see where there's some weakness in that structure, and I do think that is being difficult. But having said that, people need to remember also that when you get meeting minutes or responses, those are recommendations only. And again, if you provide additional information later, say in a submission that helps to correct the agency's view or is additionally supportive, that can be adequate. And so just because an agency has told you something here, you may say, oh, okay, I get it. They missed that. We'll fix that later going forward. So there are those opportunities as well. They're as detailed as they can be. They're based on their experience with similar products. So they have that advantage. While you may feel like you're the only person that's ever come to them with these questions, that's not true. And so they're feeding you information that's based on a broader experience or background.
Great. So, stepping back, I wonder if we can get your big picture perspective. Obviously we're talking about a number of different emerging therapeutic modalities with gene therapy, gene editing, cell therapy. Which of these has the least burdensome regulatory pathway to commercialization due to its associated CMC issues?
That's a great question. I personally, this is my own personal view, I think CMC is resolvable in any of these contexts. I think probably, let's talk about cell therapy. I think the biggest difficulty, while a cell is a complicated structure for which there's a lot of things that need to be controlled to the manufacturing process, and understanding the breadth of what it takes to control that is a big ask. I think the more difficult piece to this is cell therapies, at least nascent native cell therapies that are unmodified in any way, haven't been terribly effective. And I think that's a bigger stumbling block for development than the CMC pieces. So I'm going to put cell therapies as, I'm going to call it, least burdensome from a CMC perspective, because there's very good informational subject matter expertise. The agency, they understand cell therapies, they understand cell therapy manufacturing. They've had over 20 plus years of experience in this space. There may be nuances that are product specific, but you can address those, and they can help you with that in a way that's productive. I think the next probably least burdensome place, because of the success, would be in the areas of gene therapy where there's a lot of where there's been in the CAR T space and doing that kind of manipulation. I think that's almost become, I'm sorry to say it, in some ways, it's almost become like a well-characterized product, because the ways that these materials are produced, the way that they're delivered, and the attributes about them for this particular class of materials, very well understood, and becomes simply a case of conducting the clinical exercise to demonstrate effectiveness. So I think in that place, for certain aspects of certain types of gene therapies, there can be not as great a hurdle, because there's a lot of information. Now, AAV, for example, is a well understood manufactured process, but it has some intrinsic variables to it that make that makes some of the CMC questions around the profile of the material, and here maybe the capsid composition or other aspects about the partial folds and folds and what that material is. Those are the challenges that remain in that space. But to actually, up to that point, the manufacturer of an AAV vector is almost by rote. There's only certain ways you can do that. Same with lentivectors. So gene therapy has some values there. They're almost becoming, I don't want to quite use the word, but platform technologies in that sense. And so probably that burden of CMC wise is not as great. I think where the greatest burden probably CMC is in the gene editing place, because of its novelty, its understanding of what the tools are that are doing the editing, how do we understand what needs to be controlled in producing those materials, and then what needs to be done in the characterization space to demonstrate the accuracy of the targeted edits, the non-targeted edits, and sort of the impact of what that editing does overall within the genome of the recipient material. So I think I would sort of say probably cell therapy is a less burdensome bar, although it can be. I'm saying that's being driven largely by clinical effectiveness, and so if you don't have when you don't have a strong clinical effect to measure, then a lot of stuff becomes more important because you're trying to control for safety and integrity. But having said that, I still think there's a lot of knowledge in cell therapy. Gene therapy has some platform that's understood, and so I would say cell therapy less than gene therapy, where there's good information, with gene editing because of its novelty and newness, probably representing the biggest challenge for the CMC space.
Okay, let me ask you a follow-up to one of your prior comments when you were discussing comparability. Let's say a sponsor does some additional work on their release assays, and they really come up with something that really tightens up the potency of the released product and a more consistent with tighter margins. And they have the data that they have. Do you think the agency's position is going to be, okay, you still need to prove that this new release product that we're going to assume is more potent? Do they still need to run a new clinical study to prove that to the agency? How does the agency view something like that?
That's a great question. And I think that it's layered by what have we done to improve, to change this assay, this potency assessment? Is it optimization, for example, of the assay? You're still basically using the same assay, but you've optimized it in a way that produces more reliable information, or have you maybe taken a component of either added a new component to a potency attribute matrix or replaced an existing component? In the latter stages, it's more that I think that's a more complex and sort of information-driven exercise than optimization. If you do optimization to improve readout, agency's not going to have. They're probably going to accept that with just an exercise of demonstrating the assay's performance, because nothing's really changed. Now, if it's tightened, to your point about tightening the assay, so if the tightening now would exclude previously assayed material that has been released, that's a little more challenging. So in other words, we're releasing materials with this kind of wide range, but now we say, oh, actually the range should be this, and now you have products that you released that fell outside of that. In a perfect world, you would have retention samples where you would have retained material, understood their stability, and then you could use those retention samples, test them against the new release criteria, and find that, oh yeah, in fact, they do meet, they do fall within that range. Because then if they don't, and you've tightened the range, and you release material on a different range, it really brings into question the reliability of previously released material. So that would be kind of a struggle issue there. So it depends on the nature of the change and the degree to which, is it new? Is it a new assay component, or is it simply optimization without changing the sort of the range? But if it's a change of range, what are the implications of that? So it becomes a overall global conversation. I think the worst case scenario is clinical. I think an intermediate place to go is if you have a really good proof of concept in vitro assay or animal model that you can test against, and look at the impact of the change and make some judgments about that. It's not always the case that a clinical study or clinical data would be necessary to do that. But that's going to be, again, depending on complexity, depending on the nature of the change, the impact of that overall change, and how the agency views the significance. And that's a conversation and a dialogue. And so you need to get into that early, particularly introduce them to what you plan to do, tell them what the likely implications of that are, and what in their view is going to be acceptable for introducing that change, depending on their stage of development. Later stage, more complicated; earlier stage, less complicated.
Great. Geulah, let me hand the mic back to you for any follow-up questions you might have.
I guess just in regard to something you mentioned earlier, when therapies are becoming more potentially platform oriented. For example, if a company is developing a suite of products that are based on a similar manufacturing process, to what extent are regulators comfortable referring back to earlier data, where perhaps the main difference is the sequence of a transgene that's been incorporated into a product? What is the kind of procedure like there?
Yeah, I think it's becoming a question of interest, obviously, particularly if you're a vector manufacturer and you're simply inserting a new transgene into the construct for delivery. And so obviously, up until that point, everything is operating similarly. And so I just was at a conference on viral clearance and viral safety. And it's for recombinant therapeutic proteins is the space for which this is largely has grown up. But there they have this concept, of sort of a claim making what they call making a modular claim. So what do I mean by that? So they've got a manufacturing process for which everything is routinely done over and over and over again. So rather than each time assessing that for integrity and improving, in this case, viral clearance or from that manufacturing process, they say, when you do these steps, you've verified and validated that these steps will always get you to this point of this material. So you don't have to go back and demonstrate that every time. So to your point, if there's a platform, you could almost bring this idea of modular claiming in. So we do it the same way, with the same materials, in the same expectations, the same testing, at least up to certain points of the transgene that's being introduced. And so all of that should just be, should be good. And so what happens then is you still have, once you've introduced the transgene, you still have all of those, and you still need to provide assurance that you have to do a readout of the gene, that it was inserted correctly. And then you have to demonstrate infectivity and expression. Those things aren't going to change because they're unique, A to the gene, B to the cells that are transduced, and C to the indication. So some parts of it will always have to be done, on a case-by-case basis. But there could be upfront parts of it that could become "quote unquote" modular, or it could be simply pro forma. Typically, the way that's done now by any individual sponsor, right, is that they simply they could cross-reference their own material, or they get cross-references. All of this is done through a cross-reference mechanism. So we're not ourselves going to do all of this, but we're just going to rely on information provided by somebody else in a cross-reference approach. But in the end, at the BLA stage, you still are obligated to put that material in its integrity in your own application. So you'd still have to have some record of having either done it or having written it all out to do it. You can't cross-reference your way to success that way. So, but I think there are conversations, particularly about how it's mostly, I'm hearing it in the gene therapy space and in vector manufacturing and like you suggested. I think there are opportunities for that, and those opportunities would require education of the agency regarding that. There's this a mechanism for that, the CAT mechanism, and I can't remember exactly what the CAT acronym stands for, but it's a way that CBER, Center for Biology in particular, likes to engage early with innovative changes being made in manufacturing space, and what the implications for that might be. They don't typically have specific regulatory conversations, but how might this be regulatorily impactful? They will consider, as well as looking at your novel technology. So those are ways to do that. But I think that there is interest in platforming. Yeah.
Great, thanks. So we've just about reached the end of our time. And so in closing, I just wanted to ask if you have any final advice for development stage companies that are focused on cell and gene therapies.
Yeah, remember your timelines are too short. Remember the agency works deliberately. Make sure you have conversations at every opportunity. You're going to want to do that. Recognize that change will be hard. It's always going to be hard. When you have in product success, things you want to consider are durability of the effect, persistence of the effect. If you're in the gene therapy vector space, are there ways to cloak? Are there ways to make that not immunogenic? So you want to vector, vector novel strategies and technologies for improving their receptivity, their tropism, they're not being rejected. Remember, in some spaces you can't redose. And so maybe if we can get to a vector that allows for redosing, that would be a great step forward. And finally, looking at, I think one of the biggest things, particularly in whether it's cell or gene therapy, if it's in an orphan disease or rare space, do you actually have a really reliable clinical surrogate or biomarker that's recognized by the agency as being acceptable for evidence of clinical benefit that you can then bank on and rely on going forward in development? And then remember, don't cut corners on your CMC. Plan ahead and do risk analysis and assessment of what the impacts are going to be, and share those analyses with the agency to keep them educated with you. Those are all things I've seen as being productive and could be helpful going forward.
Fantastic. So I think that brings us to the end of our discussion. Again, Dr. Fink, I'd like to thank you for your insights and comments on the case.
Been a pleasure. I've enjoyed it. Thank you too as well.